Thanks so much for the warm introduction. Are you sure you hear me and I don't need the mic? So today we are going to delve in the science of what we talked about yesterday. And I realized that I gave this an integrated measuring modeling approach, but actually only at the end I'll be talking or just telling you again that we have produced that model. The entire talk is more on the measuring part and how we came to that understanding through the several experiments that we did. Just as a reminder, since today I told the students should always start talking from the relevance. Okay. Just as a reminder, wherever we do something in science, it's very important that today we say, why do we care? And again, for soil, we care because soils are at the nexus of solving some of our major challenges that we are facing today. And so it's not anymore a future thing, it's really a today thing that we need to be prepared to address those challenges. And since the soil organic matter really, as I told you yesterday, is able to provide many of the ecosystem services that are crucial for us to address those challenges, it's very important that we go deep in the understanding of soil organic matter so that we can inform decision makers and policy makers. And so today, as I said, we'll go deeper in the physics of the process and understanding how does indeed soil organic matter form and get stabilized so it can persist. As I anticipated you yesterday, when I started, and I guess some of my age today was given out, right? I'm 50, and I did my PhD between 91 and 94. And at that time, that is what we believed, that soil organic matter formed from the condensation product of the leftover of plant decomposition. So decomposition was basically conceived as a loss process. We were very, very interested in this decay dynamics. And so basically the understanding was that for any amount of plant residues that was left on the soil, microbes decomposed it and mineralized the carbon, so there was loss to the atmosphere. And then there was a fraction that was remaining, and we thought that that fraction was what built the soil organic matter. And the method we used to measure that, and believe me, I think I made hundreds, if not thousands, of those litter bags, was to basically put the plant material inside a nylon mesh, and you were a good PhD student if you know how to sew, because you had to sew all those mesh bags, and then you would deposit them most often on the soil surface. There was very little appreciation at the time for root decomposition. Some of us went more in depth and started putting those litter bags vertically in the soil to get a root decomposition. But basically the idea was it's very important to get the decay dynamic, the amount of loss, the amount of remaining. Now, if with the, as a PhD student, that's how I was trained to study litter decomposition, carbon formation, and something kind of didn't really ring a bell to me, because what I was thinking is, when we eat, unfortunately, we don't breathe out everything we eat, but something kind of get biomass, right? We put some weight on that somewhere has to go somewhere. That's also what I was thinking about the microbes. It's not only about a catabolic process, it's also an anabolic process and formation of other mass. And also, the thing is that I was working in forests and we were measuring this big remaining, but every other year you wouldn't walk in some more litter on the top of the soil. They had to go somewhere. And so I started thinking, okay, maybe this method is not really the best method to address all the question, because actually some of the carbon loss is not only mineralized, but is lost as dissolved organic carbon into the soil. All of you drink tea, The color in your water is the dissolved organic carbon that every plant tissue has in it and can make up to 20% of the biomass, and that actually wash in the soil at the first rain or soon after that. And that's a precious substance that microbes really like to eat. Further, the carbon remaining actually remains because we trapped it into litter bags, and in the natural environment, that particulate organic matter fragments is moved by the soil fauna and the earthworms and moves into the soil in a fraction that we can separate because it's lighter. We call it the light fraction. And the light fraction is an extension of the particulate organic matter that I was talking to you yesterday. So particulate organic matter can be light or can be heavier if it's been more microbially processed. So basically now we have two ways in which organic matter from the plant can come into the soil and then in the soil form organic matter. And I think what that's important is because the soil environment is different from the surface environment. The climate is different, the organism is different, minerals are more in proximity, and so it seemed that this litter bag approach wasn't the right approach. And so we started saying, okay, if we wanted to measure both respiration and DOC formation as well as the residue that remain, what could be as a first laboratory experiment a way to go? And so with my student Janice Sung, we thought of a system where there was an incubation where we could both do DOC and respiration. And we started off testing these 6 substrates: pine, blue stem, oak, ash, alfalfa, and actually pyrogenic organic matter, which had a very contrasting chemistry in terms of stoichiometry, in terms of carbon chemistry, so how much was soluble versus how much was structural. For the pyrogenic organic matter, even more, it's very, very highly condensed aromatic. And what we were hoping was for a nice linear regression between CO2 and DOC, so that even if DOC had not been studied in many studies, we could apply that regression from literature and estimate how much DOC was coming from, either from CO2 or maybe from decay rates. And what we find out was that it was nothing like that. So the black, oh, you don't see, sorry. I was trying to use the black point are the CO2 emission and the light are the DOC. And as you can see, there is not really any consistent trends between one liter to the other, nor with the mass loss that the liter had. And when we look deeper to understand what was controlling those DOC dynamics, we find out that actually the majority of the DOC came out within 15 days from incubation and was very strongly linearly related to the amount of hot water extractable in the plant material that we had analyzed. Later on, there was much less coming out and was actually inversely related to what we call the lignocellulose index, which is a ratio between celluloses and lignin. So it was pretty clear to us that this dissolved organic matter was a significant fraction of the mass loss, and it was dependent on the initial chemistry of the plant material. And so many of you might have read a great book, which is the Chapin Terrestrial Ecosystem book, and in the chapter on litter decomposition, that is the picture you see. And that's a great representation of the fact that the solubles are lost very quickly, the cellulose and cellulose later on has those decomposed microbial products produced, and then the lignin and the microbial products, we still call it humus, but basically this is the POM that is created at the end. So what is missing in this picture? Where does that go? We don't know where basically 80% of the plant material goes. And what's the assumption? I guess the assumption is that it's entirely mineralized to CO2. And so what we proposed is that we had to move beyond that mass loss concept and decay rate concept and start wondering where the carbon is lost to and in which form, because from plant material, carbon can be lost to the atmosphere in the form of CO2. Of course, there is also volatile organic carbon, which is another, it's very little, but is also another very understudied concept. And in reduced environment, it would be methane. But then there is also some part that all goes to the soil, some as dissolved organic matter, and some as particulate organic matter. And so we set on an expedition to kind of understand the fate of this material as it decomposed, how it formed soil organic matter. At the time when we were thinking those things, again, the picture had become more complex, and now it's not just this condensation process, but actually the formation and persistence of organic carbon was suggested to be an ecosystem property determined by the microbial transformation as efficiency, microbial inhibition because of climatic inhibition, sorption-desorption process of minerals, aggregation, and so forth. So the picture had become more complex to deal with. And in this context, in this intellectual environment, and in particular, what helped me was the fact that I was coming from a litter decomposition perspective, and I started appreciating the soil and the soil organic matter, and realized that the scientists that were studying litter decomposition were not necessarily studying soil organic matter dynamics. That was mostly studied in ag, where litter was basically residue that they dumped inside the soil. Maybe they crashed before start. So it wasn't that interesting, the process of decomposition. It was more interesting, the process of soil organic matter formation. And so being in the middle of these two scientific groups, I decided that it makes sense to have a unifying framework that could link the decomposition to the organic matter formation. And I already told you yesterday that through this framework, and in particular through the lens of the microbial transformation and the interaction with the minerals, we started reasoning on the fact that mineral-associated organic matter might form. The hypothesis that I proposed using this framework was that through the decomposition of labile compounds, so something that would produce a lot of dissolved organic matter, low carbon to nitrogen ratio, high solubles, low lignocellulose index, would be acquired and used with high use efficiency from the microbe, produce a lot of microbial and plant product, and in soils with high mineral surfaces would produce high meum, whereas the recalcitrant plant debris would basically produce particulate organic matter through only partial decomposition, and that particulate organic matter would stay there if the microbial decomposition was largely inhibited. So when you see something like that, it immediately shows you that in order to address that question, you basically need a tracer. And that's why I started being interested in isotopes in order to trace my carbon and nitrogen has moved from the plant material into the soil. And so, you know, when I was in Italy, we had an early version of a labeling, and we tried this trick also with different combination of natural abundance. How many of you don't understand isotopes? Great. What I did as soon as I arrived at CSU was actually in my first proposal to have the money to build a labeling chamber, and I realized only now how fortunate I'd been to have my first proposal being funded by NSF and gave me the money to actually build. This is a pretty big chamber. It is about 2 meters tall because we also wanted to do big grasses and trees. And it's a chamber that we actually describe, if you are ever interested to build one, in the Journal of Visualized Experiment. There is a video that describes all how it works and how you can build one. This chamber allows for an entirely controlled atmosphere in which to grow your plants. So we start from pots that have only inert material, synthetic clay, sand, and vermiculite, so that all the carbon, the nitrogen, and the water that the plant biomass is made of is controlled by us. And so we basically enrich our Ogland solution with 15 nitrogen, and we enrich the CO2 that ended the chamber with 13C. In order to not make this experiment too expensive, we have generally enriched about 7 atom percent 15N and 4 atom percent C, but you can 13C, but you can do whatever you want. Since I'm mostly interested in decomposition, we generally grow the plants, and then when they are in maturity, we kill them and take the shoots and roots and use for either lab or field incubation. When we work in the field, we try to put this material in its natural environment at the contact with so if it's root, it would go in the soil. If it's shoot, it would be up on in the litter layer, so that it's free to move and to do whatever they want. We have also experimented with producing leachate, exudate, with a lysimeter in the pot, and then we use the leachate to do some other priming studies to understand if the root exudates might prime decomposition. The other thing I will tell you I'm excited about is the fact that not all the plant material come in as normal residues. If an ecosystem is burned, the plant material come in as pyrogenic organic matter. And so I've always also played around with burning, and that is like, you have to really trust your muffle oven, because it's a $10,000 plant material that goes in, and you hope that something comes out. But we have been successful in producing dual-label pyrogenic organic matter, and also done experiment to contrast the fate of pyrogenic versus non-decomposed litter. And most recently, I got interested in mycorrhizal, and so we have also developed a technique where we grow mycorrhiza inside a mesh bag, and then we can freeze dry it and do experiment with the enriched mycorrhizal tissue. After we place all this material either in the field or the lab, there are several things that you can do with it. The quickest and dirtiest is just to analyze your bulk soil and see how much is remaining there. Or you can go deeper in your question, and you have understood by now that I'm obsessed with soil organic matter pools. And so what generally I do, we fractionate the bulk soil into different pools and analyze each of them to trace where our carbon is gone. We have done gas measurements, so you can do 13 CO2 analysis or 15 nitrogen N2O analysis to see how much of your tracer has been emitted as greenhouse gases. You can start doing some compound-specific analysis and extract biomarkers of microbes like PLFAs or amino sugars, and run on GC compound RMS to understand exactly in which microorganism used the carbon or the nitrogen that you have added. The NMR comes much nicer because you have spiked the system, and so you have much better peaks. And the other things that we started doing through a collaboration with Germany, with Carsten Müller and the group of Ingrid Kugenknabel, is NanoSIMS, where the nice thing about NanoSIMS is that you can really spatially locate on a clay mineral where your carbon or your nitrogen is ending up. We always use a mixing model approach to determine how much of our original plant material has gone in these tissues. So all the experiments that I will tell you from now on have mostly used this approach. The first one was in the Konza Prairie. And so we grew Andropogon gerardi, a grass litter, and then we added it to this color and followed it. So this was the 100% litter carbon that we put one day in 2010. And then for 3 years following, we periodically went back to the site, collected all the litter that was still on the litter layer inside the collar, and that would be that green bar. And then we also sampled the soil underneath and checked how much we found. As I told you yesterday, very much surprisingly, through the initial fast phase of decomposition, we had the most of the acquisition of carbon. Not much was acquired during the intermediate phase, despite the litter mass continued. And then a final flush was at the end. And as I was saying, in 3 years, all the litter that wasn't, of course, in litter bags had disappeared from the soil surface. We also fractionated those soils. We measured the 13C in the PLFAs. And what we found, again, was that and we also measured the NMR in the litter layer remaining and mid-infrared and acid detergent fiber method, and putting all that information together, we find out that in the first 6 months to 12 months, the litter lost the solubles and lost the non-lignin encrusted celluloses and hemicelluloses. And those we found signal of high incorporation in the PLFAs and also in the amino sugars accumulated over time. And then we found the formation of mineral-associated organic matter very earlier on in the silt and clay fraction. Only later, when lignin was lost, we started seeing the formation of the light fraction. And putting together that information, I propose that soil organic matter forms to these two distinct pathways. Again, this was a new concept because we've always thought that actually plant debris come in as POM, and through the decomposition of POM, the SOM get formed. So this is the novelty with respect to the current understanding that actually the mineral associated forms later through the decomposition of the POM residues. After that, we actually started doing this in several different systems, and through a collaboration with colleagues in Brisbane, Australia, Ellen Mitchell did the same kind of experiment, but in her case, she was interested in different addition rates, and also she has this mixed system. So what she did was not just place the litter on top of the grassland, but to simulate disturbance, like if you wanted to till that system and incorporate the litter inside the soil. And what was very, very interesting to see was, first of all, a confirmation of the fact that the mineral-associated organic matter formed early in the process before the particulate organic matter, and also stayed more or less unchanged through time. But in the mix, it basically accumulated almost twice as much as the one that was put in the surface, both in the meum and in the POM. So something seemed to happen if the decomposition doesn't occur on the surface of the soil, but occur in the inside the soil. It goes faster and is more efficient. There is more carbon that remain in the soil. So this information start giving us a confirmation of the idea that the soluble compound would form MEOM, and that the structural compound would form the light fraction and the POM. And so, you know, I like to play around with my geekiness. And I thought, what's a way that within the same plant, if I really wanted to test it, I wanted to have the same leaf material that had differentially labeled the metabolic component versus the structural component, so that I could identify in the soil what carbon was coming from the metabolic and what carbon was coming from the structure. I use term metabolic to say this soluble material. Of course, to do that, I needed two isotopes because it becomes a triple isotopic mixing model. But also, I had to differentially enrich those two substances. So the way I thought was, if I first grow my plants in the labeling chamber so that all their structures are formed, and then I move them out of the chamber for a few weeks, then their metabolic component would actually start accumulating the carbon from the natural environment. And so the metabolic component would be less enriched than the structural component, and so I could do my mixing model. That worked. We produced plant material where the hot water extractable was 2 atoms% and the structural was 4 atoms%. And then we incubated that material in the laboratory and followed the respiration and the formation of structural organic matter. And what we found out was that so this is the incubation where we measure the respiration from either the total liter, the component coming from the structural, and the component coming from the metabolic. If I zoomed in the first few days, the majority of the very beginning came from the metabolic fraction, but then actually the majority of the respiration came from the structural. So the structural did contribute to the respiration of the microbes. After 1 year, when we harvested, when we harvested those jars, what actually we found out was that all the mineral fraction, sand size, silt size, and clay size that contained the microbially transformed material were actually all only had the metabolic signal. We didn't see any trace of the structural signal in those pools, whereas when we looked at the life fraction, we found a proportion of it coming from the structural. The triple mixing model is kind of tricky, and some values came negative. But there was the most important thing was that there was no structural contribution to the MEOM, and the only contribution to the POM, the life fraction, came from the structural. So that was really interesting. Then at that point, we had become more and more, wanted then to understand, okay, is there a difference in the mineral then and the silt and clay? And so a PhD student at the time, now my postdoc, Jocelyn Lavallee, decided to do an experiment in which we did a slurry incubation to accelerate and speed up the process. But in each of these slurry, she either put shoots or roots that were contrasting for their chemistry. So the roots were more recalcitrant than the shoots, and she mixed them either with silt or with clay. She also did temperature analysis and incubated them at different temperature. But what was very, very interesting, actually, surprisingly, we didn't see any difference between the minerals. And I think that the reason is because the surfaces were anyway very far from being saturated. And so we couldn't see any difference between the silt and the clay. But everything that was produced from the shoots had more mayum formation and less CO2, and the inverse was for the roots. So basically, again, a confirmation that the labile material produced more efficient mayum than the recalcitrant material. And those were more like the geeky experiment in the lab. But then to get to my, to the most extreme of this information, one thing that we were discussing with Bonnie that we really want to get to the earth of is this mayum new formation? Is it replacing old organic matter and is a substitution? Or what happened once this new carbon gets on the mayum? Does it stay there for long, or can the microbe access it? So one thing that we had noticed in the 3 years field experiment in Kansas was that when we looked at the relation between the liter-derived carbon and nitrogen stabilized on the clay fraction, they actually tended to decrease over time. And so it seemed like some of the carbon that had been originally accumulated after 6 months might have gone. So we said, okay, how can we look more in depth in what might be happening? And at that time, I thought, let's try NanoSIMS, also because it seemed very cool thing to do. The geekiness in us like to do this stuff. So what is NanoSIMS? NanoSIMS is a very cool method. It's a humongous instrument that basically you choose a nanoscale area on your clay and you bombard it with lasers so that you then shoot into the RMS the element that come out of that area. Basically, you can map your isotopic composition on an area of a clay. As I said, we did that in collaboration with Carsten Müller at Munich. And what was nice, because I'm a quantitative person, and so a lot of these new methods are a hard thing for me because they are hardly quantitative. But what was nice, the first thing I did was I compared the isotope the 13C atom percent produced from the NanoSIM and the one I'd measured for that same fraction with the RMS, and actually they came pretty equal. So that was reassuring that this was a good method. And what we analyzed was a clay fraction from each of the time sequence that we followed in the field. And what we observed, and again, the limit of this method is that it's so expensive that you can only do one sample per time series, at least with the resources we had. And what we saw was that in the 7-month period, of course, there was the majority of the enrichment was, the logic I used here was if I have only 13C enrichment, it might just be plant material like glucose or, carbon plant material that go there. If microbial microbial products need to have the carbon and nitrogen together, co-located in the same position. And then if you only have 15 nitrogen and you don't have the 13C associated with that, then it must be a microbial product that respired off the carbon and maintained the nitrogen. So what we found was that in the 7th month interval, the majority was just carbon. In the 18 months, you start to have a diversification, and actually the majority was co-located. And finally, at the end, the majority was just nitrogen alone. So with some fantasy, and I accept that, we proposed that maybe what was going on is that at the beginning you have your tea coming out from the litter and eating straight the minerals and stabilizing as 13C rich compound. At the same time, in the litter, the microbes are metabolizing and are forming those processes where they maintain both the carbon and nitrogen from the litter, and then maybe microbial metabolite stabilize on the clay and might displace off the original carbon. Finally, over time, maybe the microbes manage to chew some of the carbon off and respire some of the carbon off so that basically the nitrogen remain accumulated. Again, this is just a speculation, but it might be a start-off model to start thinking about dynamics of this mineral-associated organic matter, in particular with respect with the possibility that some organic matter might have a preferential sorption and displace other, and also the microbe might be chewing on some of that. And the reason is because we said it is stable, but actually when they do this visualization, a lot of microbes are bonded to the mineral, and so they must be eating whatever is there. So that's the kind of some of the idea that we are moving forward with more work. And what I'm excited about is that actually through national and international collaboration, we are now conducting this experiment in a lot of different places. As I said, we started off with this grassland, and I'll show you the fire arthropods project. We did the Australian project. We then went, and I'll show you some data in the Arctic. We have an experiment going on in French Guiana. We are analyzing some of this stuff in Canada. A new experiment we have in collaboration with the UK is in Scotland. And we are doing a lot of agricultural management experiment in our sites. And so what we have something actually in Iceland in a natural warming transect, and a tree biodiversity experiment in the Netherlands so far. And we have project to start this in marshes and so forth. So it's growing and growing. And we have so far been able to produce basically everything in the labeling chamber. And our next and I have the greatest, so I have the worst green thumb. I wouldn't be able to produce any plant material anywhere. But fortunately, I have an amazing research associate, and now our next challenge is mangrove. We need to see if we manage to grow mangrove in the labeling chamber. So what I'm going to present now is some of these experiments. They are all ongoing. And so I just give you a short overview of the preliminary experiments that are coming out from some of the one where we have some preliminary experiment. One is actually at ARDEC that is our experimental research station out of Fort Collins. And this is a project funded by DOE RPE with the idea that breeding for years have been only done to increase productivity. And now with the urgency of carbon sequestration, they are looking at breeding crops also to increase roots tissue and root contribution, in particularly at depth, and how that might resolve in carbon sequestration. And so we are, it's a big team of people with, humongous crop breeding experts and phenotyping experts. And so what we did was they gave us 4 maize lines that are supposed to be 4 extremes for their root to shoot productivity. And they have this weird name, Z something. And of the 4 line, we grew all of them in the labeling chamber, and then we selected the 2 that were most contrasting. Here I just show you carbon to nitrogen. This is the root tissue, and this is the soluble material that we use as proxy for exudates. And you can see that one variety had much higher carbon to nitrogen ratio than the other. The same, they also had different lignin concentrations and different root to shoot ratios. So what we did was an experiment where in the field, here we are interested to see at depth what happened. And so what we built was 90 centimeter long PVC tube. You have heard me saying that it's very important that the decomposition happen in the natural environment, that actually roots are present when the decomposition happen. And so our tubes have always holes so that water and new roots can come in and try to establish inside the PVC as natural as possible an environment. What we did, we first went to the site with the giddings, extracted all the soils, and then we repacked the tubes so that in some treatment, the roots or the exudate, we only had exudate to do the topsoil, or we incubated in the middle, or we incubated in the bottom. And then, of course, we always have a control color where there was nothing. This is the experiment as it was a setup. And then, of course, we planted corn, and so it could live as close as possible as a corn site. We are now to the 5th month sampling, and these are the results for bulk soil of the exudate 1 month after. So this is the nitrogen remaining from the exudate that were incubated in the 0 to 20, but we also checked for them at depth to see if any carbon had moved. And actually 1 month after the addition of this very labile component, 50% of the nitrogen was there, and about 30% of the carbon was there. The nitrogen is somewhat more mobile. The carbon, we had very little down the profile. And so this will be a 2 years experiment, and we'll be continue monitoring that. This experiment actually followed the same approach that we had used before for another experiment where we were here now interested in biofuels and the possibility that sorghum would be this amazing grass that could be used for biofuel production at the same time as generate carbon sequestration with their amazing deep roots. And so the question was, is it true? Can those roots generate a lot of carbon, and how does that differ from the leaf litter? And so in this experiment done with the same approach at RDEC, but comparing the leaf tissue versus the root tissue of sorghum, and sorghum is an amazingly soft material, had a lot of solubles and relatively little lignin in both of them, but somewhat more, so the root was somewhat more recalcitrant. And what you see is that this bright green thing, which is the SOM formation, the bulk SOM formation, basically was about 10 to 20% from the beginning, and it remained. And after 3 years, everything got lost. Nitrogen always remained in more amounts. Then my student was I don't like aggregates too much. To me, it's a huge amount of work. But this student, she loved aggregates. She needed to understand everything about aggregates. And so despite I told her that she would burn out, she decided to go all the way to do the big fractionation, where basically she first separated by size the macro aggregate, the micro aggregate, and the silt and clay fraction. And then she broke the macro aggregate and isolated the micro aggregate inside micro aggregate, and so forth. So she had those 3 aggregate size fraction, and then within them, she separated the POM, the life fraction, and the MEM. So this weird colors here is all malm, but is either free when it's brown, is inside the macro, what is it? Inside microaggregate, free microaggregate inside macroaggregate, or just macroaggregate. So of this very confusing picture, what the bottom line is that actually aggregates are not a place where organic matter is protected, but it's a place where organic matter is formed. Very little organic matter is formed free. Everything happened inside aggregates. And in fact, for the one of you that did go to the site, to a field and collect roots, you see that the aggregates are actually on the material. And so all this processing happened within aggregates. If you have a loamy soil, or, if you have a very sandy soil, it's not the case. But any soil that has some aggregation, actually aggregates are a very active site for soil organic matter formation. And so while I was joking that, I don't like aggregates, actually I'm getting more and more interested in understanding aggregate as place for efficient organic matter formation rather than a place where nothing happened and organic matter stay there forever. The only place where that might be is the very, very fine aggregates. What we call the silt and clay fraction, or the malm fraction, has some very fine clay aggregates, and that's where real stabilization might happen. Quickly, one last thing that is really exciting me, and that might start becoming very controversial, is that we are seeing that actually tilling facilitate efficient formation of soil organic matter. So we have a couple of experiments in which we disturb the site and introduce the material inside the disturbed soil, and what we are finding out, this is respiration now. We don't have yet the data on the soil organic matter. So the green is the tilled one where the litter was inside the soil, and the blue is the no-tilled one where the litter was in the surface. And basically, at the cumulative is slightly less from the mix than from the surface addition, even when the surface was disturbed. And so it seemed like you have this immediate, faster flush of CO2, but then afterwards, the inside the soil, the litter stabilized, where on the top of the soil, it keeps respiring as water comes in. This is a dry system similar to here, with irrigated corn. And what is getting even more exciting is that when you complete the full greenhouse gas balance, so this is again back in Australia, where Ellen Mitchell did the same thing in 3 different texture, clay, loam, and sand, and she measured not just the respiration, but also the priming and the N2O emission. And this is a greenhouse gas balance, so negative means sequestration and positive means emission to the atmosphere. And what she found was that the mix, basically, particularly in clay soil, transformed what was a net emission into a net sink. And in the sand or the loamy, a small sink into a much larger sink. So this idea that disturbing and breaking up the system might induce carbon losses become challenged. I don't want to say that it's not the case, but it's something that is really intriguing. And further, the different responses of different textures, we are examining it more in detail along a texture and climate gradient in Canada, where we are seeing that litter from 5 months to 5 years, basically the silt and clay formation remain the same, while the palm disappear and there isn't much more formation. So again, a confirmation that that's not the case. I think I've taken too much time here. We are doing experiment in the Arctic, basically asking a very similar question across in different climate change system, and we verified that the birch the birch encroachment might actually be stimulating carbon sequestration in the soil. And another area that we are active is wildfire, and again, how the pyrogenic organic matter might behave differently than the original plant material. And so we did exactly the same thing, but not with litter, but with pyrogenic matter. And we find out that the pyrogenic matter contributes very little to the respiration, contributed very little to the assimilation in microbes and nematodes, and we found all the pyrogenic organic matter in the life fraction. You can see the difference in the axis label here. The majority of it was in the pine fraction. And the other interesting thing about the pyrogenic organic matter is that interact with the sock and the fresh carbon. In particular, when it's fresh, it might suppress some of the native respiration. I'll jump to this. I've told you yesterday that we have incorporated this understanding into this model, and I'm very, very excited now to be able in all these experiments to use this model to test some of our hypotheses and verify them. And actually, this is a picture that my postdoc sent yesterday via email for a discussion we are having. But this is how we are moving forward. We want to have the root inputs and also separating the microbial in a bacterial and in a fungal pool, and we are adding the nitrogen. One last thing I want to say is that it's not just about us scientists. I think that one thing that I really enjoy doing is going to school, and my students do the same. And I feel that in particular when we live in a community that doesn't appreciate us that much, we need to start when they are young. And in particular in elementary schools, they don't have, the teachers are not trained to be scientists, and they wouldn't do any science if we don't go there and excite the kids about that. And so this is, we partner with the Puder District School, and we do a lot of outreach activity in the classes, and it's really, really, really fun. I jumped to conclusion, and the last thing I want to say is that at Colorado State University, I really have the fortune to be one of several soil ecologists and soil scientists. And so every summer, we offer a 2-week summer soil institute that is really, now we are almost at the 10th year, I think, this year. And we go to the field, we teach a lot of different methods. And it's really good for students that need a full immersion in soil, and maybe they don't have that in the institution where they're going, or they're changing field. They were trained in a different field, and now they have to do soils and just want to get it all in one time. So if any of you is interested, we offer it every summer. Just contact me or go on the webpage. Sorry for being so long. And when I talk about the things I like, I think I talk too much. Thank you. Any questions?
I was wondering about some of the priming experiments that you did. Do you see evidence of priming? Like, I'm only really familiar with it in like an aquatic context.
Yeah. Yeah, so we did several priming experiments. In the majority of the case, we did see priming. So we are actually submitting soon a paper that we say walled soils. Every soils that we tested prime, so the addition of fresh substrate induced the emission of carbon from the native soil. If you do the budget tool, that carbon emission is never more than the amount of carbon that remain in the soil. So the question that I want to get into is it true priming? Is it a substitution so that the fresh carbon displace some of the old carbon from the mineral associated that get respired. So I think it's a combination of the two. But I am against the big thing about priming because not many of the priming study do the full budget. And the majority of the case, when you do the full budget, the priming is not more than the addition. And actually, there is a net sink. So I think that priming needs to be discussed with caution and need to be discussed with the full budget. Does it make sense?
When you were talking about the dual label study, and then you use that to sort of to explain this trend over time of the label nitrogen ending up in the mineral fraction more than the carbon. I think I understood that. So
Both the carbon and nitrogen.
Both the carbon, but did you say that you think eventually microbes are able to pick off the carbon?
Yeah, so the NanoSIMS work wasn't done in the the dual labeling of carbon and nitrogen, not the differential labeling. Yeah, I think that some of the carbon that goes in on those minerals might be respired off by the microbes. But again, that experiment, I now go in 2020, would be 10 years, and we'll go back. And so we have the ability to see 10 years later if there is still that much carbon in the silt and clay fraction. For sure, in the Canadian sites, where we fractionated their soils. So this is an experiment that I didn't establish. This is at Gregoridge, but they never fractionated it. So I got all the soil to fractionate. And the silt and clay didn't change. From 5 months to 5 years, it didn't change. So it might stop, it might be stable. Or it depends on how much saturated maybe it is. And so when it's oversaturated, there might be more of that displacement. And when it's not saturated, it might stay there. But there isn't much of a trend with the texture. We have to dig more in there. I showed you a graph that actually my students sent. I haven't yet. I wouldn't present the data that way. But it seems like can stay, particularly maybe if the minerals are not saturated.
You were talking about we had a figure showing how you fractionated the carbon into different pools. Is it all physically fractionated or is it chemically?
Yeah, so the fractionation scheme I showed yesterday. What we do was we first disperse everything. So right now, the way in which I'm doing it, I consider aggregates a state in which the primary fraction are present. So I either measure aggregate as a state of the soil to do the mean weight diameter. But when I do the fraction, I first disperse everything, and we shake with beads. So we don't do ultrasonic dispersion, otherwise the carbon moves from one fraction to another. So we disperse with beads, and then by centrifugation we get the dissolved organic matter. Then we float off in 1.85 gram per centimeter cube using SPT the light fraction, and then we collect the dense fraction and we wet sieve it at 53 to separate the silt and clay that I call MAM from the sand-sized palm, the heavy palm. And the nomenclature of the fraction have changed through my publication. Now I try to call everything light palm or heavy palm, the light fraction versus the sand-sized palm, and then the silt and clay. In my early study, I separated it in silt and clay. Then I start seeing no difference, and so right now I'm keeping it together. We are doing some study to understand. The male makes about 50% of the carbon in a lot of our soil, so we believe that we should go farther, but we haven't yet found what's the meaningful fractionation to go after the 50. So we are trying hydrolyzing the carbon or doing some silt and clay. So what is challenging is that you find the new carbon everywhere. And so this idea that there is this very, very old persistent carbon is hard, because if we found our new isotope, even in the non-hydrolyzable fraction that has a 14C age of, several thousand years, it's like, what's happening? So that's the big dilemma to me, to find the old fraction that doesn't contain the new carbon. That's what we are searching right now in my lab. Okay, thanks so much.